Welcome to the MOOC NPTEL course on proteins and gel-based proteomics. Before we move on to gel-based proteomics and discuss various techniques and concepts involved in proteomics, let me give you an overview of whole course. In week 1, we will talk about the basics of amino acid, and then we'll move on to the structural level of proteins, their folding and misfolding. Further, we'll study various chromatography techniques employed for the protein purification. In week 1, the first lecture will start with basics of amino acid, and then we'll discuss about the structural backbone and their classification. Amino acids are the building blocks or monomers that make up proteins. They consist of a central alpha carbon atom bonded covalently to an amino group, a carboxyl group, a hydrogen atom, and a variable side chain called as R group. Amino acids are the basic monomeric constituents of proteins found in varying amount depending upon the type of protein. They are classified based on the properties of their side chains or R groups, which vary in size, structure and charge. Polarity of side chain is one of the main bases for the classification. Amino acids having nonpolar aliphatic side chains including glycine, alanine, proline, valine, leucine, isoleucine and methionine. We will then discuss about different type of amino acid isomers, L and D isomers, with respect to the conformation stability and their natural propensity to be found in the L isomeric form. So, amino acids have 4 different groups which are connected to the alpha carbon atom. It can form 2 mirror images which can exist in L or the D isomers, which are shown in the slide here. The alpha amino acids are chiral. There could be R or S configurations in the amino acid depending upon the priority groups, but only L amino acids are present in the proteins. All the L amino acids have S configuration, which describes the counterclockwise direction as shown here from highest to the lowest priority groups, which is an indicative of chiral center with the S configurations. Some of the isomerism properties of amino acids will be discussed in following animation. Before learning about the isomerism, let us first know what is chirality. The term chirality arises from the Greek term chair, meaning handedness. Just like the two hands are non-superimposable mirror images of each other, amino acid molecules are also non-superimposable due to their chiral alpha carbon center. All amino acids except glycine contain an asymmetric center that makes them chiral in nature, due to which they can rotate the plane of polarized light. The 2 enantiomers designated as D and L rotate the plane of polarization in opposite directions. The 2 enantiomers of amino acid are non-superimposable mirror images due to the spatial arrangements of 4 different groups about the chiral carbon atom. Rotation of either isomer about its central axis will never give rise to the other isomeric structure. In next lecture, we will talk about the versatility of proteins as macromolecules and the building blocks of body. Further, we will scrutinize the structural hierarchy of the proteins. We will talk about secondary structural elements commonly found in proteins. Understanding different levels of structural organization of proteins can help us to extrapolate their functional significance to the cell or organism, and it will be covered in this lecture. In third lecture, we will talk about the tertiary structure of proteins, which is formed due to folding of proteins. We will also discuss about the improper protein folding and aggregation, which can be corrected by the molecular chaperones, which facilitates the process of folding in case of large proteins. I will also discuss about the significance of protein folding with respect to diseases and functions like cellular trafficking, regulation of cell cycle, and cell growth. The next lecture will be focused on protein purification, peptide isolation, and analysis. We will discuss in detail about different types of chromatographic techniques like gel filtration, ion exchange chromatography, affinity chromatography, HPLC, strong cation exchange, and reverse phase chromatography. These chromatography techniques are often used for various proteomic applications, and it will be good to cover their basic understanding in the first week of this course. The lecture 5 will explore the field of proteomics and give an introduction to proteomics, how it is different from conventional protein chemistry. In this lecture, we will discuss about how proteomics can provide a holistic overview of studying the proteins and protein complexes. We will discuss about how protein chemistry and proteomics differ in the techniques which are employed in these approaches, as well as their scale of usability. The traditional protein chemistry has employed some of these techniques like electrophoresis and mass spectrometry. So what is different? The advancement of these techniques, along with the genome sequence availability, has made that difference, which will be discussed in this lecture. The sensitivity, resolution, and robustness of high throughput approaches, which has made this transition, will be discussed in detail. The second week will focus on systems biology and proteomics, as well as the sample preparation strategies. The first lecture will start from systems biology examination of a biological entity as an integrated system rather than studying its individual characteristic reactions and components. The various ingredients of system biology. For example, if you are studying about a cell and a system behavior, one need to look at the genome, transcriptome, and proteome profile, how protein DNA and different type of transcriptional networks are altered, protein-protein signaling networks, multimeric complexes, how are they formed, and how protein is localized in the intracellular dynamics and metabolic networks. All of these are ingredients of studying about a system. There are several techniques which have been used in proteomics, which typically aim to elucidate the expression, localization, interaction, and cellular function of proteins. Lecture 2 will provide an overview of various gel-based techniques such as SDS page, two-dimensional electrophoresis, and dye etch, which are commonly used for proteomics applications. Next, we'll talk about the sample preparation strategy for proteomic analysis. The sample preparation is very essential for any proteomic analysis. The protein extraction methods aim to extract all the proteins in a cell or its organelle, and the presence of interfering components have to be minimized. Different biological samples pose different challenges. For instance, serum proteomic analysis is very complex because there are different proteins of different order of magnitude present in serum. Some are very highly abundant and some are very low abundant. Proteins such as albumin and immunoglobulins are highly abundant, and it is preferred to remove some of these abundant proteins prior to any proteomic analysis. In bacterial protein sample preparation, sonication becomes very important to disrupt the bacterial membrane. The sonication process breaks open the cellular membranes to release the intracellular components. Accurate protein quantification becomes very crucial for the quantitative proteomic analysis. Protein quantification is sensitive to detergents and certain oils. Therefore, it becomes crucial to select the correct quantification method. Absorbance of standard and unknown protein samples can be measured, and protein concentration can be determined from the standard plot of absorbance values. Serum proteome analysis. In this interactive animation, I'll show you stepwise how to collect serum and how to perform the proteome sample preparation. So, first click on the syringe so that blood can be collected. The blood proteome is one of the most complex component of the human proteome. It fluctuates depending on the physiological and pathological conditions of the patient. So, collect almost 5 ml of blood from the vein of healthy or diseased participants using serum separation tubes. Immediately after the blood collection, the tube should be kept in ice for 30 minutes for clotting. After clotting, the sample are centrifuged at 12,000 rpm at 20 degrees for 10 minutes. Serum can be separated and multiple aliquots can be made for further storage at -80 degrees. So, during this animation for the sample preparation, I will describe one method which we are currently using in my laboratory, where we have tried combining different type of methods being available from different published protocols. So, the crude serum can be diluted 5 times with phosphate buffer pH 7.4 . Ensure that mixing is uniform, and then vortex by clicking on this vortexer here for 30 seconds. Mild sonication is performed by using a sonicator for 6 cycles of 5-second pulse with 30-second gap in between at 20% of amplitude. The mild sonication step in serum sample is found to be effective in improving the gel quality and resolution. Now, you can start depletion strategies to remove the abundant proteins. There are commercially available depletion columns. The affinity binders or ligands are immobilized to a solid support, the chromatographic medium, and used to specifically bind abundant proteins from a complex protein solution. The depletion strategy effectively enhances the resolution and provides the maximum number of spots on two-dimensional gels, thereby a better coverage of the whole serum proteome. The depleted serum is mixed with ice cold acetone containing 10% TCA, and after vortexing, the mixture is incubated at minus 20 degrees for protein precipitation, followed by centrifugation. After incubation, the tubes can be centrifuged at 12,000 rpm for 15 minutes at 4 degrees. The pellet can be dissolved in lysis buffer containing 8 molar urea, 2 molar thiourea, 4% chaps, 2% IPG buffer, 40 millimolar of DTT and trace amount of bromophenol blue. You can then again perform the vortexing step and keep this extract in minus 80 degrees until further use. Fifth lecture of week 2 will focus on SDS page gel, where proteins get separated on the basis of their molecular weight. The low molecular weight proteins having high mobility migrate further through the gel, and high molecular weight proteins remain close to the point of application. Week 3 will be entirely focused on two-dimensional electrophoresis principle, methodology and challenges involved in performing good 2D gels as well as various applications. In two-dimensional electrophoresis, the first dimension separation occurs based on the isoelectric point of the proteins, and second dimension separation occurs based on the molecular weight using SDS page. Isoelectric focusing works on the principle of isoelectric point, which is pH at which the net charge of protein is 0. Two-dimensional electrophoresis remains one of the core technologies to study the proteome, and it is applied for various biological applications. It is relatively easy, an affordable technique, and it's highly sensitive method to visualize the proteins, which makes it widely used methods for protein separation. So in this animation, I will describe you the two-dimensional electrophoresis process. Prior to isoelectric focusing in two-dimensional electrophoresis, the commercially available IPG strips must be rehydrated. This process can be done either by the passive rehydration or active rehydration. In passive rehydration, the IPG strip is placed with its gel site downwards in a well containing the protein sample, reconstituted with a suitable buffer. As you can see in the animation, you can apply the protein sample and then add the IPG strip so that it can absorb the protein solution. This process can be done for 10 to 20 hours depending upon your length of the IPG strip. This is then covered with mineral oil to prevent the gel from drying up and left overnight. Other process known as active rehydration, where the protein sample is added to the strip via a sample cup, followed by the cover fluid to prevent the gel from drying up. The protein sample being applied, a very low voltage condition is provided and then cover fluid can be added. This is then placed in the isoelectric focusing instrument and low voltage is applied. Process can be performed from 10 to 20 hours. These loaded strips are then focused on an isoelectric focusing unit by passing the current. The various proteins of the sample mixture migrate in the electric field and come to rest when the pH is equal to their isoelectric point or Pi. So they become neutral and are no longer being affected by the electric field. The progress of electrophoresis is monitored by adding a tracking die, which you can see in the animation moving ahead of the proteins. The IPG strip is then equilibrated in a reducing agent such as DTT, followed by an alkylating agent iodoestramide, which prevents reformation of reduced bonds. This strip containing the separated proteins is then placed on SDS page gel slab for further protein separation in the second dimension based on the molecular weight. The proteins on the IPG strip are subjected to SDS page by applying a direct current between 100 to 350 volt depending upon the size of the gel. Any protein that may have been present as a single band on the IPG strip, as you can see in the red circle, due to similar isoelectric point, can then be separated based on the molecular weight, where the smaller proteins will migrate the farthest. This is view of a sample gel which has been run by two-dimensional electrophoresis and stained with Kumasi blue. Each spot provides the information about the molecular weight and isoelectric point of the protein. If your protein separation and staining is fine, then at the end you should be able to see good protein separation on gel. But many times there are different types of gel issues which could give rise to very bad gel image. These issues could be because of the sample preparation method, reagents involved in 2D process, isoelectric focusing, various parameters and different type of staining methods being used. I'll also show you different substandard or bad gels and what could be the possible reasons behind them, which can help us in troubleshooting these kind of artifacts. Some of these issues of gel-based proteomics can be overcome by using an advanced method which is known as two-dimensional difference gel electrophoresis or DIGE. The two-dimensional electrophoresis can be used for a large number of biological applications. These include studying the global protein expression, which is the separation and identification of all the proteins present in a given sample, comparison of protein abundance between the test and control samples. This is one of the very common application, also known as differential protein expression or abundance-based proteomics. Resolution of proteins from the complex mixture is separated using 2DE. Studying different type of isoforms or the post-translational modifications is another application which is commonly used. One of the major advantages of this technique is the visual analysis of the proteins on gel, which is not possible from the gel-free approaches such as mass spectrometry. In the last module, in week 4, we will talk about a more advanced and sensitive gel-based technique known as two-dimensional fluorescence difference gel electrophoresis or 2D DIGE. There are 3 different Psi dyes, Psi2, Psi3 and Psi5, which are used for the DIGE experiments. The excitation and emission wavelength of these 3 are defined and used during the scanning of the gels. In this lecture, we'll talk about 2 different types of labeling strategies, lysine labeling or cysteine labeling. Lysine labeling method is also known as minimum labeling method, where the charge and the size of dyes are matched. When to use which type of labeling method and the rationale of using these labels will be discussed in this lecture. In next lecture, we'll talk about overall steps involved in performing DIGE. While most of the steps are exactly same as what we have learned in two-dimensional electrophoresis, but major variation will be in the preparation of internal standard. In internal standard, all your control and treatment samples will be mixed together and it will be separated on one IPG strip. The DIGE gels can be scanned using a fluorescent scanner such as DIGE imager, and Psi3, Psi2 and Psi5 patterns can be obtained. After scanning, from same gel, 3 separate images are generated. Further, a combined image from mixing all the 3 dyes and the overlapped images are generated. In overlapped image, the red and green are indicative of overexpression or underexpression of a given protein, and the white or yellow color represent that there is no differential response. The DIGE image analysis is very robust and automated as compared to the two-dimensional electrophoresis analysis.
Yes, as we were talking about the 3 samples we can label with Psi2, Psi3, Psi5. Now you have 2 different samples from different source. Now you are taking these 3 samples into 3 different Eppendorf tubes and you are labeling with Psi3 dye as well as again another with Psi5. Then you are taking those 2 samples, pooling them and labeling with Psi2. Then you're mixing those 3 into a single tube, then you are running in a single strip. There you will be analyzed in first dimension the IEF, whereas in first dimension after analyzing IEF you'll be placing into second dimension, where you are completely analyzed the second dimension. Now this gel can be scanned in the Typhoon Trioplus, where you will have 3 images from a single gel. Then this can be analyzed with the help of decider software, then where the complete analysis can be done with the help of decider. Then in DIA there will be a co-detection. The co-detection nothing but there is a standard gel which is labeled with Site2. From the same area we are expanding remaining 2 images also, so that the artifact can be minimized. Again, this is fully automated analysis. There will be very less manual interference would be there in this analysis. And after finishing of DIA, we can go to BVA. In BVA, we can compare all DIAs together. Like as you can see in this video, this complete first of all it matches between the standard gel, and from standard gel to again the corresponding DIA. It matches each and individual spot, and it will give you the final data. Then these BVAs can be analyzed in the further in EDA, that is extended data analysis. Here you will have lots of stringent statistical data, that's a differential expression analysis, pattern analysis, discriminant analysis, and PCA analysis.
Once you have identified the protein of interest using gels, you would like to identify them using mass spectrometry. So the last 2 lectures of this course will focus on MALDI-based mass spectrometry. The mass spectrometry has the ability to provide the platform for comprehensive coverage of proteome, and it has become a very important analytical tool in biology in general and in proteomics during the last decade. In this lecture, we'll talk about the concepts of MALDI, how the time of flight analyzer can be used to resolve the ions produced by ionization source on the basis of their mass to charge ratio. Briefly, we'll also talk about the data analysis and protein identification using mascot. Overall, this course will provide you the basic knowledge of gel-based proteomic techniques with a special emphasis on two-dimensional electrophoresis and difference in electrophoresis or DIGE. Hope this information and knowledge acquired from this course will be useful to you, whether or not you work in proteomics research, but these techniques can be used for several applications. I hope this course will be a stimulating experience for you. Thank you.